Good afternoon, my name is Yase, and this summer I worked at the Kalume lab at Seattle Children's Research Institute. My project studies the effect of sleep deprivation on epilepsy in a mouse model of Megalencephaly. How do you go with this? Okay. Oh. So, megalencephaly is a developmental disorder associated with brain overgrowth, frequent cortical malformation, and variable intellectual disability and epilepsy. The Dobbins lab at SCRI has recently identified that gain-of-function mutation in PIK3CA gene constitutes an important cause for MEG. PIK3CA, the Kalume, Dobbin, and Milam lab at SCRI have developed a mouse model which recapitulates all the human features of MEG. PIK3CA is a gene in the mTOR signaling pathway and is very well known to be involved in various types of cancer. Figure on the top shows that MEG mouse model have larger brains compared to the mutants, and the figures on the bottom show that they're from a coronal section of the brain, showing that the mutant shows an enlarged cortical compared to the control. So the PIK3CA mouse model develops epilepsy, and on the left side of the panel, the mutant shows epileptic features. We can see regional spikes and myoclonic seizures. And on the right side, this is an example of a mouse model that was sleep deprived, and it shows generalized tonic-clonic seizures. On the top panel, there's a left and right EEG and EMG recordings, and the bottom panel shows enlarged view of seizure onset on panel A and seizure end on panel B. The Kalume lab is interested in sleep in the context of epilepsy because sleep disturbances are common in people with epilepsy. And sleep disturbances are associated with poor seizure control and poor quality of life for patients. Poor sleep can lead to behavioral and attentional problems, which can ultimately contribute to social and academic difficulties in their life later on. Now I'm going to talk about the experimental procedure, and the experimental procedure is that the mice used in these experiments were held under 12-hour EEG electrodes were implanted using techniques developed in this lab. The electrodes were placed bilaterally through small cranial burr holes over frontal and posterior cortices and were fixed in places. EMG electrodes were placed underneath the neck muscle, and a reference electrode was placed at the midline cerebellum. So there were two groups in this experiment, sham and sleep deprivation. Both groups were subjected to sham and sleep deprivation for 5 hours, and post-sham and post-SD was recorded after that period. In the sleep deprivation group, mice were kept awake for 5 hours at the beginning of a light period by gentle touch with the white curtains in that little chamber, and the curtains were controlled by the lab chart software macro and power lab to move randomly in different patterns as using pulse waves as illustrated above. The sham groups were also placed in the same chamber, but they were not subjected to the gentle movement of the curtain and thus were allowed to sleep. In a different chamber, we recorded EEG and EMG activities and video of two groups of mice. And through the power lab data acquisition, we were able to convert from analog to a digital signal. Through a software program, we're able to further process the raw data through certain filters to extract right and left EEG recordings as well as EMG signals. The data that I used to analyze was from the post-shaman and the post-SD. The figures on the left are examples of spikes in a Megma's model, and interictal spikes are defined. These are interictal spikes, and interictal spikes are defined as high amplitude, sharply contoured EEG waveform and no association with movement on video. The figure on the right shows the average number of interictal spikes between post-sham and sham deprivation group. Error bars indicate the standard error of the mean, and there were only 3 animals per group. In total, we found that the post-sleep deprivation group produced a higher number of interictal spikes, and in conclusion, sleep deprivation leads to an increase in the number of interictal spikes in Meg mice, and this finding indicates that sleep deprivation exacerbates the severity of epilepsy. Interictal spikes are the hallmarks of epileptic brain, and in clinics they help to confirm the diagnosis of epilepsy and characterize generalized and partial epilepsies. In future, I would like to increase the number of animals in order to further this experiment to have more data, further characterize the impact of sleep deprivation on other markers of epilepsy such as myoclonic seizures, generalized tonic-clonic seizures, fast EEG activity, and focal spiking. And I would also like to examine the changes in epileptic events in relation with non-REM sleep and REM sleep. And special thanks to Dr. and Mrs. Special thanks to Dr. and Mrs. Ellenbogen, my lab, Dr. Kalume, Dr. Dolby, Michelle Bard, Arena, Shannon, and Thomas, and also special thanks to the University of Washington Department of Neurological Surgery faculty. Thank you so much.